So, without further ado, I'd really like to welcome our panelists for today. So we've got two speakers who are going to talk to you about their individual pieces of work. First up, we've got Dr. Paul Potter, who is a group leader for disease mechanisms and aging at Oxford Brookes University. Paul has a long-standing interest in this field, looking at the pathogenesis of disease and understanding the genetic basis, and then translating that into improving the models that we use for this. Over the last few years, Paul's been focusing on the relationship between aging and disease susceptibility, particularly in age-related and chronic disorders. He is a member of the Arthritis UK Centre in Oxford. He's an honorary senior lecturer at Imperial College as well, and a member of the Royal Society of Biology. So we're very much looking forward to Paul's presentation. And second up, we have Dr. Satomi Miwa, who is a postdoctoral researcher at the University of Newcastle. Satomi started her career in exercise science, but became very interested in energy, metabolism, and mitochondria. She studied work in that field specific to aging at Cambridge University before joining the team over in Newcastle. Satomi and Thomas von Zielinski are one of the recipients of our Proof of Concept Awards and have a specific project around this. Satomi has very kindly narrated this on one of the e-posters, so please do take a look at that. And now, without further ado, I'd like to ask Paul to pop his microphone on, and I will be handing over to Paul. Thank you very much.
Thank you very much for the kind introduction, and I'd like to thank the organisers for an extremely flattering invitation to start this off. So, as I'm on the warm-up act, I'll crack on. So I started this talk and it started to turn into a list of models of aging, and it got quite boring. So what I've tried to do is bring together a lot of factors that influence study design, how we measure things. So if there's one thing you take home from this lecture, it's this: the best model of a cat is a cat, preferably the same cat. And I apologise if I overuse this at all, but it's a really important concept to get across. We're dealing with models. There are a lot of mutants that tell us lots of interesting things about biology. Some of those mutants model some disease to a certain extent. It's very rare you get a model that will recapitulate everything you see in the human population. It's nigh on impossible. So how and why do we measure aging? Basically, we wanted to distinguish between people that are aging healthily and those that need interventions or assistance. We need to know the consequences of our interventions, and we need to quantify their effects. Lifespan has been used to a great extent, but it's limited in what information you can gain from it. And in terms of modelling disease, is there really any scientific benefit in letting mice reach end of life and become morbid? We want to intervene and study disease before that stage. Health span is now taking over as the main concept. It's how long your health lasts, but it's quite difficult to define this. It involves a lot of constant assessment of your physiological parameters. It's quite easy to do in model organisms, but with patients it's particularly hard. So the translation is quite difficult. So there are other things we can look at in aging. Frailty is something we all instantly recognize. When you talk about frailty, people understand it immediately, but how do you measure it? How do you quantify it? It's defined as an accumulation of deficits. There isn't a standardized model as yet, but there are increasingly commonly used indices. There's the Fried phenotypic model, which defines frailty as an accumulation of 5 areas of phenotypic five phenotypic criteria: weakness, slowness, low levels of activity, self-reported exhaustion, and unintentional weight loss. Another very useful index is the Rockwood Frailty Index, which is slightly more complicated, but still very applicable. It assesses frailty according to these levels. There's a lot of criteria associated with this index, but they're all quite easy to measure, and they're all quite quick in assessments. And the most important thing is, when we're talking about models, we need to be able to translate the data from model organisms to humans and vice versa. Frailty is very useful. It's been used in longitudinal studies, frailty index. It's been shown to predict adverse outcomes after surgery, adverse outcomes just in normal living, and as I mentioned, it's applicable to animal research as well, a preclinical model. So it's a good translatable system. And here we see some data from Rockwood. You can see with older mice, they accumulate frailty phenotypes, and the index is the summation of all these phenotypes. But most importantly, this is very translatable into human. You can see here the overlap between the human of frailty indices and the mouse frailty index to normalized age. So that we now have a useful measure of aging that's quantifiable and relatively easy to measure. It's been successfully applied to studies. Mice love running on wheels, so this is looking at exercise and intervention. And the real point I want to highlight is they looked at frailty indices and they found benefits in younger mice or young adult mice. The frailty scores improved after exercise. This was already also seen in old mice, an improvement. But they also did some more detailed studies, and it shows one limitation of the index. You can see here grip strength improved as an average, walking speed improved, endurance score improved. If you look at these box mice, Q7 and Q8, they really came from the quite severely frail level up and improved greatly. However, not everything improved. So although frailty indices are useful in an overall measurement and translatable, just using index alone, you may lose some of the detail in the data. Related to frailty is multimorbidity. It's the presence of two or more conditions. It's a big impact, but the key thing about multimorbidity is it seems to occur before frailty. Most frail patients are multimorbid, have a number of diseases, but multimorbid patients are not necessarily frail. So it looks like frailty occurs after multimorbidity. So we're moving down the aging spectrum and measuring something a bit earlier. Unfortunately, there's no pattern of disease we can measure. There are some associations between different diseases. You can measure it by the simple accumulation of the number of diseases. In patients, there's a number of multimorbidity indices that have been applied, similar to the frailty indices, but I'm not really aware of much in the way of translation of these multimorbid indices. But there may be some amenable factors like clinical biochemistry results that would be translatable easily from mice to patients and back again. Resilience is another factor that is of interest in measuring in aging. So frailty, as I said, is an accumulation of deficits, and these deficits are thought to occur because of a reduction in resilience, the ability of your physiology to respond to stressors. And this declines with aging. It may be related to the accumulation of diseases and multimorbidity. Multimorbidity may be a positive feedback in the reduction of resilience. So if there are homeostatic measurements, mechanisms you can measure, they're very useful. There's the potential to assess responses to stressors, such as infection, thermal challenges, simple circadian rhythm challenges. They're amenable in mice, but not necessarily so amenable in patients. But it's something of interest that people are developing protocols in. So what other markers of aging have we got? epigenetics is clearly something useful. We know sirtuins have an influence on aging and their deacetylases. There are clearly transgenerational effects on aging that I'll come back to in a minute. There's measurements of an epigenetic clock which can distinguish between chronological and biological age, and this seems to be predictive of some outcomes. In terms of simple biomarkers, there hasn't really been a great deal of success. There are things like you can measure levels of senescence and telomere shortening, but in patients, you can't take multiple biopsies across tissues and start looking at cellular senescence. So they may be useful and are being used, but the translatability is limited. So we know rapamycin extends lifespan, and the reason there's logic to bringing this up. So we have a range of effects, improved lifespan, some improved health, but this is really to emphasize the importance of measuring many parameters. The paper by Neff et al. Looked at the effects of rapamycin, but they tested young and old mice. It found that there were limited effects on many aging parameters, but what they found was some of the effects that were beneficial also were beneficial in young mice. There were a few parameters that specifically improved in old mice, and that they concluded that lifespan extension may simply be reduced cancer incidence because of the effects of rapamycin on cancer. So there's obviously a lot of debate about these results and discussion, but the key thing I wanted to get across is the advantage of a wide range of phenotypic tests, but also testing different ages and the depth of data you can get from that, rather than just looking at lifespan or end of life aging phenotypes. So in terms of what we want from a model of aging, what do we want? We want a rapid development in aging. We don't want to wait around. We want all the phenotypes that are associated with aging. We want a reproducible phenotype with low variance, so we can get smaller cohort numbers. We also want to capture the stochastic effects of aging, the different diseases occurring in different orders in multimorbidity. We want to reproduce the variability seen in patients, so things like epigenetics. We want the models to be amenable to high throughput testing or have nice biomarkers. They want to translate all the phenotypes across all ages and all environments. We want clear windows where we can intervene and then study the outcomes. In short, we want mice to do everything. So good luck with that one. It's a big ask. So we'll focus on a few things. Senescence, obviously associated with aging, clear effects, and again, this emphasizes the link with senescence. You can there are senescent cells accumulating in aged tissues. You can induce disease by implanting senescent cells. There's a whole load of data out there, but again, what I wanted to do was emphasize this paper where they implanted senescent cells in young mice and induced physical dysfunction. The greater the number of cells they implanted, increased more physical dysfunction. But more importantly, they were looking at a particular pathway. So not studying aging per se, they are looking at the effects of senescent cells and the effect of removing them. And when they delete senescent cells, they get a good outcome. So here's just a bit of data: increasing levels of senescent cells, a decrease in physical parameters, but again, not everything decreases. So this again emphasizes the need to do a wide range of phenotyping tests, not just rely on Frailty index. Doing the reverse and taking out senescent cells, again, you see improvements, but the improvements are variable. Not everything is significantly improved. There's downregulation in inflammatory markers, but an extension in lifespan. So really, this is just emphasizing that again, I think you need a lot of detailed studies. There's no simple readout of aging. You can't have a simple answer to a complex question. Some mice models are very interesting and very useful. There's Progeria mice that result in aging phenotypes due to the accumulation of mitochondrial mutation. They have a reduced lifespan. Some of the interventions provide beneficial effect. There's some new papers coming out on the links between mitochondria and senescence and multimorbidity, and the contribution of mitochondria to the full senescence phenotype. So we've got to see how that all relates back to what we're seeing in these mice. There's Progeria models. The concept is simple. You want fast answers to aging, so you look at models that have accelerated aging. There are a number of interesting models, Hutchinson-Gifford syndrome models. There are other models with all various aspects of aging, linked to phenotypes associated with aging. I think we're still away from determining whether it's true aging, but these are models that can answer specific questions about specific pathways. The one downside is that, again, known interventions known to improve aging are improve phenotypes in these mice. The one downside is these mice are accelerated. So, are we dealing with a worst case scenario? We're looking in a model that has a really bad accelerated aging, so we may miss subtle effects of interventions. Beyond models and how we phenotype them, there's a whole load of metadata that we need to consider before you even start planning. Breeding and phenotyping, what are your outcomes, what your intervention points. This is an interesting point. As I said, there's quite a variation in data. So do you go by chronological age or by an aging parameter as a time to intervene? Protocols have got to be standardized. Then we can start comparing results much more easily. Age affects some of the phenotyping protocols. So I've run old mice on gait analysis, and they really don't want to run. So there's that to consider. Whether you look at cross-sectional cohorts or longitudinal studies, because some phenotyping tests will make you can't test the mice repeatedly. Welfare checks are incredibly important. You've got to talk to the cage side staff. They are experts in this area and really pull out mice that are outliers. Weights we found to be very good, particularly when you can start detecting rapid weight loss, but there's a lot of time involved in weighing mice continually. And just to highlight this is a screen I did on aged mice. Basically, the point to emphasize is a lot of phenotypes and a lot of mice were picked up just by simple expert cage side staff. So the communication between the research and the cage side staff is important. Background data, there's a lot of data out there that you can get hold of. There are strain and sex effects. I really want to put this in to emphasize the choice of strain. I have come across reviewing something where they were arguing for using a strain that had a shorter lifespan because it was easier to study and it cost less. But that particular strain of mice suffered from a particular cancer. So you weren't looking at accelerated aging or a shorter lifespan, you were looking at increased cancer. So understanding the foibles of the inbred strains you're using. They all have oddities. Black 6 mice go deaf, they have NNT mutation that affects metabolism and so on. There's lots of data out there, but you need to be aware of your mice strain. We found, and I think most people find, there's a lot of variation, an increased variance in data from old mice. And this has an impact on increased cohort sizes to make sure you have a significant outcome. This is just a simple data showing percentage fat mass in young and old black 6 mice, and you can see the spread of data. This is a very carefully controlled colony of mice, as well controlled as you can get, and you can see in an inbred strain there's a massive variation in the fat mass, and that we see that across a lot of phenotypic parameters. So when you're setting up breeding, there are factors that influence that. It's clear there are epigenetic factors affecting metabolic phenotype. So the metabolic status of the dams affect lifespan and metabolic phenotypes in the offspring. The age of the fathers have also been shown to be important in a lifespan and testing. And also the environmental consideration. This is often overlooked. Mice are pretty lazy and they get quite fat. So are we looking at the best scenario here in laboratory mice? That's something else to consider. And we saw how we've seen how that affects the studies on caloric restriction in macaques. So in summary, we can assess aging at lots of different stages. And to be honest, the earlier we can assess it, the better it is. We can use biomarkers, surrogates, but we still need, I think there's still the argument to do a lot of in-depth phenotypic analysis of both young and old mice to look at interventions. We need standardized protocols. There are lots more models. Some of them are very good at answering very specific questions in specific pathways, but we may need to look at different models if we want to look at aging generally. And there are lots of considerations long before you start your experiment. Point of intervention, how the breeding is done, the status of the parents. And I would just like to thank everybody involved in the Mouse Age Cost Action, because these were a lot of discussions we had. It was run by Ilaria, who I think you probably know, and they were a great bunch of people. Thank you very much. That's it.
Thank you very much, Paul. And if you've any burning questions, write them now if you worry about forgetting them. And while you do that, I'd like to introduce Satomi, who is going to give the second presentation today, which will pick up on some of Paul's excellent points around senescence and models. So Satomi, over to you.
The aim of my talk is to present some examples of our work using mouse models to ask our research questions. The research questions we have been addressing are centered around the role of cellular senescence in aging, and specifically, one, how do senescent cells accumulate during aging? And two, can anti-senescence interventions slow down the aging process in the long term? So we are addressing specific questions. We used two contrasting environmental treatments to manipulate lifespan. One is extension of lifespan by dietary restriction. And the opposite is accelerated aging by whole body irradiation, by radiotherapy. Paul has already mentioned, but here is a quick background of cellular senescence. Cell can become senesced by telomere attrition, DNA damage, or other forms of stress, such as oxidative stress, oncogene. And senescent cells are different, they are larger, they have mitochondrial dysfunction, they produce high amount of pro-inflammatory cytokines and reactive oxygen species. And these phenotypes are collectively called the senescence-associated secretory phenotype, SASP. The problem of these cells are that because of SASP, they can influence surrounding cells to make them senesced. And then senescent cells spread. We have learned the in vivo relevance of senescent cells. We found senescence phenotype are also found in post-traumatic cells, such as neurons and muscle. We also found senescent cells accumulated with age in different tissues in mice. Now, here's our first question we had. It was quite some time ago. How do senescent cells accumulate during aging? Does it depend on biological age? And is that reversible? And we use dietary restriction model to answer these questions. So DR, dietary restriction, is a reduction in calorie intake without malnutrition. It is a well-known intervention to be able to extend mean and maximum lifespan in a variety of model systems: mouse, rats, Drosophila, C. elegans, rhesus monkeys, and most likely humans too. And this is our own big dietary restriction study in mice with 40% restriction of food intake. And these are two male and female ad-lib normal fed controls. And these are blue lines are males and female restricted group. And this is female. The longest lived little lady living until 43 months until she finally died. So it was very long term. DR has been shown to induce a variety of stress response pathways, such as reduction in IGF insulin signaling and toll pathway, NF-kappa B, and stimulation of AMPK, sirtuins, FOXO, NLRP2 pathways. So dietary restriction has been instrumental to understand the molecular mechanisms that regulate lifespan. Now, we wanted to see if the rate of accumulation of senescent cells was a function of biological age in each different mouse model that has different maximum lifespan. And for this, we used the dietary restricted model as well as some genetic models too. So here is a lifespan curve for ad-lib and DR, just as I showed you. And this is ICRF alpha. It is a C57 black 6 strain that is genetically long-lived and statistically longer-lived. This is a wild type versus NF-kappa B knockout mice, which has a chronically high inflammation. And this is a knockout, and this is a wild type. And here's a graph showing a maximum lifespan. And this is a third generation Turk knockout, which is a short telomere. This is NF-kappa B knockout. This is a long-lived ICRF alpha, this one. And this is ad-lib control on DR. We looked at amount of senescent cells in liver at the different ages of these mice and plotted their age as a function of maximum lifespan. So it's a normalized age. And there was a really good correlation showing senescent cell accumulation, senescent cell abundance correspond to biological age. Yeah, so the rate of senescent cell accumulation correlates with biological age in different models. So next, we wanted to see if senescent cell accumulation can be reversed by DR. So what we did was to switch the diet here. So this is a body weight. So this is ad-lib control, and body weight peaks around 600 days. And this black line is dietary restricted mice. And here, ad-lib switched the diet at 12 months for 3 months up to 15 months, and then back again. And this blue is DR, switched to ad-lib for 3 months and back. And then we looked at liver again of senescent cells. And this is a hepatocyte of a telomere-associated foci, which is a marker of senescent cells. And blue is ad-lib, goes up with age. Red is DR 3, 12, 15, and stays constant. And yellow is DR switched to ad-lib for 3 months. So this one switched to 3 months here. And they had lasting benefit of dietary restriction previously. So senescent cell was significantly lower than just a normal ad-lib control. And green is most remarkable. So these are ad-lib fed, switched to DR just for 3 months. And abundance of senescent cell back to just as if they have been DR for all life. We did something similar to immunodeficient mice, NSG mice, which lacks a functional immune system. Black is wild type, red is knockout, and the thick line is ad-lib, and dotted line is DR. So both genotype ad-lib mice had increased senescent cells, and DR and wild type DR, as you can see, show reduction in senescent cells, whereas knockout senescent cells remain the same. These studies suggested DR stops generating new senescent cells, so it makes it stop, so remain same. However, clearance of senescent cells, the reduction in senescent cell, as you can see in wild type, depending on active immune function. Now it's back to here. recently, the approaches to selectively kill senescent cells have been developed called Synalytics, senescence lysin, and it stops spreading of senescent cells. Alternatively, xenomorphics are the ones which do not kill senescent cells but masks to prevent SASP factors so that by effect, it prevents spread of senescent cells. In fact, some well-known dietary restriction mimetics, such as rapamycin and metformin, are known as xenomorphics. Now, many studies have shown senolytic treatments improve multiple of age-associated conditions in mice. So here's a long list of various conditions are treated by different types of senolytic treatment. And here's our paper, and this is a review we have done recently. So senescent cells are apparently causal to many pathological conditions, meaning they're very important therapeutic target. And here, the second question we had, can anti-senescence interventions slow down the aging process in the long term? Long term is important because many studies didn't look at long term. They just finished the study, killed the mice quite quickly afterwards and rushed to publication, I guess. And particularly, we used Synalytics and metformin, which is, as I said, xenomorphic and dietary restriction mimetics. We used the model of accelerated aging, which is caused by radiotherapy. And we used the whole body irradiation for this. There's a reason. Accelerated aging in long-term cancer survivors are a serious problem. About 1 to 2 million people in the UK, cancer survivors suffer from increased mortality, increased morbidity, and enhanced frailty. It is thought to be caused by therapy-induced senescence by DNA-damaging therapies, such as radiotherapy. So we wanted to see, can we prevent this premature aging by senescence cell targeted therapies? And we wanted to test in this mouse model, especially wanted to see if it can prevent accelerated aging, whether it can treat accelerated aging, and does it have lasting effects? So this is what we did in mouse model. For the mouse of 5 months, we gave irradiation, whole body irradiation 3 times of 3 gray. And once they have recovered next month, we gave 2 types of senolytics, as a nabitoclax or dasatinib quercetin, we call it D and Q. And then we followed up to 17 months old. Alternatively, we did irradiation to 5 months again and let them age up to 12 months by the time they have developed premature aging. Then looked at what happens afterwards. So here is a frailty index, as Paul explained. And this each dot represents each individual mouse. And here is black is non-irradiated control, and black thick line shows regression lines. And thin lines surrounded by this are 95% confidence interval. Red is irradiated control. Quite impressively, both nabitoclax and DNQ managed to normalize the rate of increase in frailty index, just like non-irradiated mice. And this case, it's even more quite remarkable. After onset of senolytic, it's a short term, just 2 weeks of senolytic treatment. Again, normalized the rate of increase in frailty index. It did not prevent, it did not reverse already developed frailty, but it's prevented future increasing frailty index. Now, this is a neuromuscular function by hanging wire test, and the pink one is a success. Success means mice managed to hang the wire for 1 minute, or put 4 limbs up to the hanging wire, or reach the end of hanging wire. And as you can see, this is 5 months after senolytic treatment, so it's about 10 months, 11 months. And both senolytic and improved performance, And this is up 10 months, so it's almost here. They still had a lasting effect of senolytic treatment given at 6 months. In this case, it's treated mice is already premature aging mice, and afterwards, at 7 months old, they had already also had improvement in hanging wire. Here is a cognitive function. So here is measured at 17 months. Both senolytic had improved cognitive function, which is measured by Y-maze test, and it tests short-term memory. And here is the same story. Again, late treatment of senolytic improved cognitive function, short-term memory. So senolytics were able to prevent and treat the markers we assessed. How about metformin? So this case, we had irradiated mice, and in 6 months, we gave metformin for 3 months, and then left it up to 17 months, just 3 months of metformin. And here is a frailty index. Just like senolytic treatment, metformin was able to normalize the rate of frailty index increases, just like non-irradiated normal mice. So metformin was also effective. Again, neuromuscular function was also improved, and straight after metformin, but long after finishing metformin treatment, they still had a benefit. And the cognitive function also, they had a tendency to improve short-term memory. We also looked at senescence markers in liver. First, we checked if irradiation caused similar changes as normal aging. So we looked at comparing young and old mice and normal unirradiated, sham, and irradiated mice. And loss of nuclear HMGB1 is a well-established senescence marker, so we looked at it. And loss of these nuclear positive cells are a sign of increased senescence cells. And the old mice are lower, and same as irradiated mice. Nuclear size is also a good marker of senescence cells, and increasing senescence cells means increasing nuclear size. And again, old mice increased nuclear size, and the same as this, and irradiation also had a higher size of nucleus. So irradiation causes similar changes in terms of senescence markers in liver. Now here is the effect of senolytics. So both senolytics prevented the reduction in nuclear positive HMGB1 cells. And same as metformin, metformin was able to prevent the reduction in these cells. And nuclear size, both senolytic and metformin prevented increases in nuclear size. So senolytic and metformin also decrease proportion of senescence cells. This is a cellular level of evidence, tissue level of evidence. Now this is interesting, skin epidermal thickness. And aging is known to cause skin thinning. And as you can see, mice, young and old mice, reduction in epidermal thickness. So this is a mean epidermal thickness. This is a photo. So it's the same magnification. And these layers are epidermal layer, and young mice has this much, and old mice is this much. However, interestingly, irradiation made an increase in epidermal thickness as a side effect of irradiation. So for epidermal thickness wise, aging decreases, but irradiation increases. So irradiation is not capturing everything what we see in aging. So this is specifically different. Now I just like to summarize what I've been showing. First question we had, how do senescence cells accumulated during aging? As a function of biological age, the model which helped were dietary restriction and genetically short and long-lived models. We also found DR can reverse senescence cell accumulation, and the model was dietary restriction crossover. We also found it was cleared by immune cells, and that was helped by doing DR on immunodeficient mice. And second question, can anti-senescence interventions slow down the aging process in the long term? And we had a model of accelerated aging by irradiation. That was a model for cancer radiotherapy. Irradiation had broad effect, cellular, physiological, and behavioral effect. However, it's not exactly the same as aging. It increased the skin thickness. We found in this model, Synalytics can prevent and treat accelerated aging parameters we measured. And it was also clear that one-off short-term Synalytic intervention had long-term lasting effects. Metformin was also effective. Next, so what we are currently interested in is a more realistic radiotherapy model, such as brain-specific irradiation as a model of brain tumor therapy. And we have a collaboration with cancer research people. We are also interested in application of senescence-targeted therapy to other conditions. And most importantly, we are very interested in a discovery of novel and safer, more effective senolytics, which is why I have an e-poster on this. And particularly, we have in vivo evidence of such senolytic, improved senolytic, and now currently testing in UK spine-funded research for this at preclinical level. And finally, I'd like to thank many people, Thomas, my supervisor, and Tom Kirkwood. And these are collaborators in our institute, and this is lab members and collaborators. And here's our lockdown lab meeting. These are funding body and CRUK Pioneer Award and UK Spine, of course, and other BBS Lucy and Shima and industry partners. And thank you very much for your attention.
Thank you very much, Satomi. That was a fantastic presentation. And I know that we've got quite a few questions coming in. So Paul, oh yes, welcome back. I wanted to start the questions with Anthony Holmes, who joins us from the NC3Rs, who wanted to make a comment on how this fits into their own work. Anthony, do we still have you?
I'm still here, Bev. Yes, thank you very much. I'll be as brief as I can because I recognise there are some great questions in there for the speakers. But thank you for the opportunity to just to have this chance to tell you a little bit more about what we do. So I appreciate that some of you will know about the organisation in some way. So I'd just like to give a very quick introduction to us. So we are the National Centre for the Replacement, Refinement and Reduction of Animals in Research. And we were established in 2004 by UK government to support the research community in applying opportunities to reduce animal use and improve animal welfare where they're still required because of the scientific benefits this brings, in addition to the ethical benefits. We fund research, early career researchers and innovation, and we also have a large program of in-house activities which are led by our staff to drive changes in policy, practice and regulations. And we work internationally with industry across the pharmaceutical, chemical, consumer and agribusiness sectors, academia, regulators and funders, and our remit is broad, including any area of animal use for research purposes across biological, medical and veterinary sciences. And our annual budget is about £10 million, and most of that comes from government through MRC and BBSRC, but we also receive funding from the pharmaceutical and chemical industries and charities to support specific programs of work. But coming back to the meeting, I think both presenters have made a very strong case for the importance of animal models in ageing research, and animals will continue to play an important role in this. But what we want to try and foster with it and support at the NC3Rs is a research community that is willing to critique the models that are being used, both in terms of their animal models and their in vitro models, to ensure that they really are the most appropriate and humane models that answer the research questions that are originally adopted for. And I'm no expert in the area of ageing, but a few examples that come to mind are all opportunities for adopting in silico, cell-based, non-mammalian models like Drosophila, C. elegans, and yeast being explored as part of an integrated approach to help inform studies done in animals and in the development of new animal models. Are there technologies available that will improve behavioural and cognitive assessment in identifying novel behavioural phenotypes associated with ageing? So, for example, home cage monitoring and automated continual trials approaches for spontaneous object recognition tasks. What are the latest advances in science that could be applied? For example, does whisker movement have some utility in quantifying motor and cognitive decline in ageing? And I think there is some evidence out there to suggest that, and there are some technologies being developed like LocoWhist to look at gait and whisker movement concurrently. And Paul alluded to this, but it's really important. I'd like to come back to it, is how important appropriate welfare monitoring is. And you might be interested to know that we are currently addressing this, or hoping to address this, through a funding competition that we have open at the moment to develop an app that uses artificial intelligence and machine learning to automatically detect changes in facial expression and body condition to improve the monitoring of mouse welfare and reduce the resource that's actually required to do this by the technicians. And this is being championed by AZ, GSK, CRUK, Manchester Institute, and the Sainsbury Wellcome Centre. And we know that there are obviously activities ongoing in the space to try and support this and do this, but there are also lots of challenges that researchers face, which means it's possibly not at the forefront of what they're doing. So it would be really wonderful for NC3Rs to work more closely with this community to make sure of the resources that we have available to you and the funding schemes that we have available to you to try and help you and support you in some of these considerations. So, for example, we have a skills and knowledge transfer award scheme to support the transfer of established models which can have a 3 Rs impact from one group to another, and this is currently open for applications with a closing date of the 27th of November for outline applications. We have a Crack It Open innovation platform to connect academia, industry and small companies in developing new and commercializable models to address specific research challenges which involve the use of animals. And also thinking about reproducibility and credibility. You know, we have a very large program of work looking at this in vivo research, and tools such as our experimental design assistant to support researchers in ensuring that the studies they're developing and designing are the most appropriate in terms of the number of animals that are being used, and that this is consistent with the scientific objectives. Reduction of bias has been considered, and that appropriate statistical methods have been applied. And then finally, once those studies have been conducted, our ARRIVE guidelines, which for reporting animal research to improve the reproducibility and make sure that the very key information is included within those publications so that people understand why you've done it, how you've done it, and then can interpret the outcomes from that. So I'll leave it there, Bev, and I'm happy to pick up with anybody after this if anybody's interested. So thank you.
Thank you very much, Anthony. And I know that you've got to dash off now, so that's why I came to you first of all. And
thank you.
Anybody that wants to pick up with Anthony, we can connect you after the session.
Thanks, Bev. Take care.
Thank you. And so back to the floor, we're getting quite a few questions in, and those that we can't answer in the time remaining, we will circulate to the speakers to get responses on them. I'm going to go to the first question that's coming up on my list. Let me just pull that up, which is from Gregor, and it's, do you have any data about the reduction of inflammation and fibrosis as primary drivers of aging? And I think that goes to Paul first and then Satomi.
Ooh, that's a good question. Yeah, I think there's people like Janet Lord that would be much better placed to answer this than me. But yeah, I think inflammation is a large part, and I think the senescent cell contribution to inflammatory drivers is really important. So, in a very brief answer, yes, I think there is data out there about how inflammation can accelerate underlying diseases. Fibrosis, not my area, I'm afraid. Really don't know. I would imagine it's part of a sort of multi-morbid phenotype that it compounds other phenotypes as well.
Yeah, as long as I know, from liver fibrosis has been reduced by both dietary restriction and Synalytics. So it's liver, but nonetheless, senescence, fibrosis is a really good evidence that they are important factor.
Thank you both. I think sometimes when questions are asked, it's sort of a question that remains open, as in this area we've still got so much work to do. So thank you both. Number two that we've got coming in from Didier is the lifespan and maximal lifespan of rats, especially, I'm not quite sure how to pronounce this rat model, the Sprognik mouse model. Where's the best place to find this information out?
Off the top of my head, lifespan for rats, I think it's Sprague-Dawley is the strain, is 3 to 4 years. I don't think there's been any sort of large-scale aging studies on rats off the top of my head. I would guess the best place to start with would be breeders, suppliers of rats, because they'll have the information on at least the age of their breeders. Unfortunately, I've worked with rats. They're much nicer than mice.
So, Satomi, anything more to add to that?
Not for this, but yeah.
So yeah, no, raising an important point about how as a community we access these resources, and again coming back to points that I think you both raised around standardisation here. So question number 3 I'll come to is from Marion, and it says, thank you ever so much, Dr. Potter, for such an interesting presentation. Is there any particular biomarker identified for the detection of CVD onset in animal or human studies?
Ooh, that's way out of my field of expertise. I think I have read papers looking at biomarkers of CVD, but I can't recall any off the top of my head, I'm afraid.
So I'll actually come in here and say that we have a session later this week, a workshop, looking at biomarkers that are predictive of multimorbidity. And although I can't comment directly on the results of the finalised study, this is a question we can take to that workshop with Carl Hennigan and Gary Ford to look at whether their data analysis has pulled any specific biomarkers out here.
Thank you very much.
Our pleasure. So moving on to question number 4, which has come to us from Chaz. If Chaz is still with us, he may want to ask the question directly. He may not still be with us. So let me just find the question. So Chaz Bantra has asked what the effect of Synalytics are in osteoarthritic joints. I open that to both of you.
I think they have, if my memory serves me well, there was a study involving Judith Campesi where they looked at the local removal of senescent cells in an osteoarthritic joint, and it improved outcomes. What the exact details are, I can't remember, but yeah, as far as I can recall, it does have a beneficial effect.
Satomi, anything to
Yeah,
add?
Yes, I agree. Yeah.
Thank you. I'm just going to keep on moving through some of the questions. So question number 5 from Claire Edwards, who may also be James Edwards. How do Synalytics target senescent cells only, and what's the criteria for a good Synalytic?
Yes, so this is very important. So the mechanistically, they tend to target upregulated survival mechanism of senescent cells, and mainly that's apoptosis resistant fatty acid, lipid metabolism. And these are the target of Synalytics. And in fact, it's a very cytotoxic, so generally they are toxic. And many of them are repurposed cancer drug, and the navitoclax especially, those things. And then related to other questions, the dose are really important. And we particularly use the lower dose of navitoclax than published data because there's a good evidence of side effects, but our studies show it's still effective. So what's important is really to evaluate what's necessary to prevent side effects of Synalytics. How good it is, we just have to try. And main criteria is specificity. Is it specific to senescent cell? Does it have effects on young healthy cells? And of course, effective, sensitivity. Yeah, so those are criteria, and we work on in vitro model to test this. Yeah.
Thank you, Satomi. You've got to switch the lights back on. We've got many more questions that have come in, and I'm looking through them, and we're going to run out of time, so we will circulate those and get some answers and pop those around to everybody. Just to close up, first of all, I'd just like to say a huge thank you to Paul and Satomi for responding to our call to action to talk about this area to open our conference. I think there's been some really, really important points raised around, you know, what do we need in these models? How are these models working? How can we make better Synalytics? And I think it's the beginning of a really interesting chapter here, or maybe the middle. And I'd just like to give Paul and Satomi a few seconds to sort of summarize their presentations today and any final remarks before we sign off for the afternoon. So Paul, to yourself first of all.
Yeah, well, thank you for the invite again, and all the questions. I'll try and answer them the best I can. I think, yes, there's some very interesting models out there. There's some very interesting ways of looking at them. The overall design for these experiments is not straightforward, and I think expecting simple answers to the questions of aging is asking a lot. We need a lot of detailed phenotyping and complicated models, I'm afraid. That's science.
Indeed, such is life, but we're not going to fall shy of the challenge. Satomi, some closing remarks from yourself.
Yeah, it was such a privilege to be able to be here, and it's really good to know more about UK Spine network. And yes, I think we need to be wise to use be able to decide which model to use for what particular questions. And Paul's talk was really helpful to help us to give a warning. Nothing is perfect. It's a model, but nonetheless, we can study how to use it best. And then I think that's really helped our research so far. So hopefully we can continue.
Thank you very much. And with those remarks that we have much more complexity and much more to do, I am going to close today's session and say thank you to the speakers and to you all for joining us. We hope to see you at sessions later on in the week. So do come along and join us, and have a lovely afternoon. Thank you very much, everyone.